Hello students, I am teacher Svetlana, and in this lecture we will learn about the cell death and the cell division, that is meiosis, which is covered in the chapter cell division. So let's begin. Cell death or death of a cell, you may think of it as a bad thing for the cells in your body to die. In many cases, that's true. It's not good for cells to die because of an injury, for example, due to scrape or any harmful chemical, which is called as necrosis. However, some cells of our body die not randomly but in a carefully controlled way. For example, during the embryonic development, the cells between the embryonic fingers die in the process called as apoptosis to give a definite shape to the fingers. This is a common form of programmed cell death where cells undergo cellular suicide when they receive certain signals. Apoptosis involves the cell death, but it benefits the organism as a whole. For instance, by letting fingers develop or by eliminating potential cancerous cells. So here is the image. It tells you apoptosis is a programmed cell death. You can see here you have the cell which is normal. Now the faulty enzymes must be digested or they can be incorporated into another cell. So this is a cell which is of no use now to the body. So here it undergoes its stages of self-destruction, and finally you have the scavenger cells which digest the apoptic bodies. So the cell is eliminated from your body. Now students, let us have a look at meiosis. The term meiosis was coined by J.B. Farmer in 1905. It takes place only in the reproductive cells during the formation of gametes. By this division, the number of chromosomes is reduced to half. Hence, it is also called as reductional division. The cells in which meiosis takes place are termed as meiocytes. Meiosis produces 4 haploid daughter cells from a diploid parent cell. So meiosis is of having 2 subtypes. We have the first meiotic division, which is also called as meiosis 1, or it is also called as heterotypic division. And then we have the second meiotic division or the homotypic division, which is called as meiosis 2. So let us have a look at the stages of your meiosis. So let us have a look at meiosis 1. In meiosis 1, it begins with your prophase 1. The prophase 1 is being further subdivided into leptothene, zygotenin, parchitinin, diplotinin, and dikinesis. Let us have a look at what happens in short in each of these stages. In leptothinin stage, the chromosome condenses and becomes visible. So you have here, you can see the nuclear membrane is there. You have the freely suspended genetic material which starts condensing in the form of chromosome. In zygotenin, chromosomes start pairing together to form homologous chromosomes by the process of association called as synapses, which is accompanied by the formation of a complex structure called as synaptonemal complex, and the complex formed by a pair of synapsed homologous chromosome called as bivalent or tetravalent. So here you have this bivalent forming. In parchitine, exchange of genetic material between the 2 homologous chromosomes. Homologous chromosomes are the chromosomes that are identical, morphologically and structurally identical chromosomes. They exchange their genetic material, and this process is known as crossing over. Takes place between the non-sister chromatids of the homologous chromosomes. Non-sister chromatids are the 2 different chromatids which belong to 2 different chromosomes. Now, why this stage is very important? This is the stage which brings about change in the genetic makeup. It alters your genetic makeup. It brings or introduces variation. Diplotinin stage is characterized by the dissolution of the synaptonemal complex, and egg-shaped structure called as chiasmata forms by the tendency of the recombined homologous chromosome of the bivalents to separate from each other except at the site of crossing over. So here you have now the synaptonemal complexes that is there, it starts to dissociate. In dikinesis, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation at the end of dikinesis. The nucleolus disappears and the nuclear envelope also breaks. So here you can see that gradually the nuclear envelope is disappearing along with the nucleolus also disappearing, and now it is ready for its next stage. Then we have the metaphase 1. The bivalent chromosomes align on the equatorial plate just as it happens in your mitosis. The microtubules from the opposite poles of the spindle attach to the pair of the homologous chromosomes. So this, if you remember, resembles your metaphase of your mitosis. So it gets arranged here at the equatorial position. Spindle fibers hold them from the center point. Then we have the anaphase. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. So here you have at the anaphase, again same, you can see that the centromeres are, to the centromeres the chromatids are pulled apart, where the centromere is held by the spindle fibers and the two chromatids are loose. Then you have telophase 1, the nuclear membrane and the nucleolus reappears. Cytokinesis follows, and this is called as a dyad of cells. The stage between the two mitotic divisions is called as interkinesis, which is followed by prophase 2. So here you have your cytokinesis 1 taking place after karyokinesis, that is after the nuclear division, cytokinesis occurs and the two haploid cells are formed. In many cases, these daughter cells pass through a short resting phase or interphase, which is also called as interkinesis. In some cases, the changes of telophase may not occur, that is, the telophasic changes do not occur, and the anaphase directly leads to prophase of meiosis 2. So from here, anaphase, in some cases these steps are skipped and directly prophase 2 occurs, in that is, it begins the meiosis stages of meiosis 2. Meiosis 2 begins with your prophase 2, it contracts to meiosis 1, meiosis 2 resemble a normal mitosis. So here in contrast, your meiosis 1 was very much different and your meiosis 2 resembles your normal mitosis. The nuclear membrane disappears by the end of prophase 2 and the chromosomes again become compact. So here you have the nucleolus disappearing, the nuclear membrane disappearing, and this chromatin material begins to condense and form compact chromosomes. At metaphase 2, again the chromosomes align at the equator and the microtubules from the opposite poles of the spindle get attached to the kinetochores of the sister chromatids. So same, it gets arranged at the metaphasic plate at the equatorial positions and the spindle fibers are holding the chromosomes at the kinetochores. Then you have anaphase 2. The centromeres of each chromosome splits, allowing them to move towards opposite poles of the cell. So here again same, there is splitting so that the two splitted chromosomes move towards the opposite poles. Telophase 2, the two groups of the chromosomes get enclosed by a nuclear envelope. So now you can see here that the two chromosomes that are there, they again get enclosed with the nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of the cell. So you can see here that cytokinesis follows and then we will have a tetrad of cells. 1, 2, 3, 4, 4 haploid daughter cells are formed. Now these undergo further changes to develop into gametes. Significance of meiosis: meiotic division produces gametes. If it is absent, the number of chromosomes would double or quadruple, resulting in the formations of monstocytes, that is, abnormal forms of organisms. The constant number of chromosomes in a given species across generation is maintained by meiosis. Because of crossing over, exchange of genetic material takes place, leading to genetic variation, which are the raw materials for evolution. And this is the reason why we humans or any organisms are not exactly alike or similar to one another. Though we belong to the same species, we show a lot of variations. Even you compare yourself with your sibling, you might show a lot of differences. Some might even not appear to be considered as, if anybody sees you, might not believe that you two are siblings or your brothers and sisters or your brothers, because there is a lot of differences between the two of you. So these differences come because of genetic variations, and this happens only because of meiosis. Thank you, students, for participating in this lecture. See you soon again with the next topic. Till then, take care, be safe, and keep learning.